using this genetic information and the clinical data that we have, we also brought in gene expression data from over 200 tissues and hundreds of individuals. And we were able to partition the genome into regions that are more highly expressed in a specific tissue compared to other tissues in the body. And then we can ask that partitioned genome, does it contribute significantly more to heritability than other tissues? And we applied this to MS susceptibility, and we saw strong enrichment in immune tissues, so in the spleen and in lymphocyte subsets, and this is expected. But then we applied the same analysis pipeline to MS severity, and we saw strong enrichment in the brain, adjusting for multiple testing, in multiple brain regions and in the cervical spinal cord. So what this is telling us is that the genetic influences, determinants of disease severity in MS are acting through regulation of gene expression in the brain and in the spinal cord. And the implication of this is that while MS starts through a peripheral immune perturbance based on genetic data that we have, the resilience to the disease process and what determines the outcome of an individual is largely the resilience of their central nervous system and biological processes that are acting within neurons and glia. We are now delving deep into the biology of this and trying to understand how these genetic variants influence gene expression and influence disease outcomes to be able to identify specific genes that we can then target with therapeutics and be able to move forward the understanding and therapeutics of progression in MS.